Good morning. I'm not super sure what I've got going on today. Let's see, I'm rerunning that gel, gotta reprocess those samples. After that, I have about 40 different cultures to transfer from our tubes to fresh tubes, which are going to be sent to University of Calgary. That's going to probably take up most of my day, so I'll try and get a little lunch lecture in for you. See you then. Dear colleagues, as you learned in the university-wide message this afternoon, the rapid evolution of the COVID-19 pandemic has required Northwestern to accelerate its response to this unprecedented challenge and to further modify research activities. As described in today's earlier memo, essential research can continue on campus. Such work is defined as COVID-19 research that has the potential to mitigate the pandemic's spread. That's us. Are you guys working on a diagnostic?
I don't know. That would be a Carly thing.
Oh, okay. I assumed you would know. So, this is everything that didn't end up soluble. So, see how nice and clear that is? Here's everything that doesn't dissolve in our buffer. So, I'm gonna try to share with you just how we do a nickel affinity purification. I know this is kind of like drinking from a fire hose, but I'm going to do my best to walk you through why it is proteins like to stick to nickel. And the real fact of the matter is, it's because I tell them to. Proteins are, of course, strings of amino acids. Amino acids are kind of like pearls on a necklace, and the necklace is a protein, right? So we add a specific sequence of amino acids at the very end of our proteins. And that sequence is H, 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 H. 1, 2, 3, 4, 5, 6. Yes, H. Histidines, coded for by H, are particularly sticky when it comes to nickel. Sometimes cobalt or other metals too. But that is what's in these columns. Nickel. Stuck to resin bead. 300. Now that our columns have been equilibrated with some buffer, we're all happy, we can add some sample and get these puppies moving. And there it is. Hopefully that material on the bottom is devoid of our target protein, and our target protein, just the one with that polyhistidine tag, 6-axis tag, it's all in that resin on top. So, because it would be completely useless to take something that we've been praying was soluble all along and then make it insoluble, we're going to use 1 molar imidazole. Imidazole is basically histidine, but without the rest of the amino acid. It's just a little ring. That made no sense, did it? But it's going to displace the histidines off of nickel resin, and that is going to make it pull back through. And now that is, with a little bit of luck, purified protein. hold on. I forgot my tweezers. Alright, here we go. 58. So, each of these cultures, each of these tubes contains a little bit of frozen bacteria. What do bacteria love to do more than anything else? Make more bacteria. But we've been able to hijack that desire in order to get them to express whatever proteins we want, because bacteria are pretty simple machines, as it turns out. We can sort of just program them to do what we want, which is like the coolest thing about them, right? So each one of these contains a different set of instructions to make a different protein. And that is what we end up using in our science. Alright, so here's what I was hoping to see. This guy is N protein. N protein's got the insoluble fraction, the soluble fraction, and then just the fraction that's stuck to our nickel resin. So that's awesome. This is the same thing. This is exactly what you hope to see. This guy is papain-like protease, and you actually see we've got a fair amount of expression right there. What is expressed is only a little bit soluble, and what's soluble doesn't really stick to the nickel column very well, at least not in this clone. This construct, however, expresses a little better, is a little better soluble, and sticks to the column just a little bit better. And you see that mirrored over here where I loaded more sample, where a little bit stuck, but a lot more stuck over here. Not quite as good as N protein, but pretty good. So I'm pretty optimistic about this. I have one more that I can test, one more clone, one more, man. Well, I'll get into what a construct means probably in another video, but I have one more type that I can test. It's got an interesting handle on it that makes it easier to work with. So we're gonna try that. Well, that's my kind of work day. That was 6.5, 7 hours of complete madness. I am completely beat. Let's see, we got N protein express. Turns out we're gonna use that to build an ELISA. More about that later. We're going to try a new approach to getting papain-like protease. I've got a few tricks before we throw in the towel and give it to Sergey, who, God, Sergey gets all the credit. Not that I'm bitter. Let's see. Like next week, we've got NSP4, NSP14. It's gonna be a big week, so yeah, I'm excited. As always, ask me any questions that you have. I'm more than happy to elucidate or obfuscate. Yeah, I'll see you next week. What have we here? Paper goods, huh?